Good morning. I want to talk to you about the MHC things again, okay? MHC1, MHC2. Now, there's a page in here on figure 2218. I alluded to that yesterday. MHC is a protein produced by a certain chromosome 6, I think. I'm not really sure about that, but that's just an educated guess. I got between 1 and 23, 1 and 22 I can choose from. But anyway, it presents a processed antigen. Whatever that the antigen is that's been engulfed, whether it's a bacteria or a virus, part of that is fragmented, part of that marries it to a lysosome, and there are smaller fragments, and those fragments are produced, picked up by the MHC proteins, and go to the cell surface, and they stick to the cell surface. They're presented on the outside of the cell. Now, MHC1 is found in all nucleated cells. All nucleated cells, okay? The MHC2 is found on the antigen-presenting cells, the monocytes, the Kupffer cells, the microglia cells, okay? Now, once this antigen is presented, a fragmented antigen is presented right here, what the hell is going to happen? That's what we're going to pick up today, all right? What is the benefit of having a processed antigen? Now, figure 22-19, antigen recognition by and activation of cytotoxic T cells. Okay, so here we have MHC1. MHC1 is going to be recognized by a T cell, not a B cell, a T cell, right? Now the T cell, just follow me along right here, the T cell fastens to the MHC1, okay? But it's not quite good enough to maintain establishment as far as the length of the attachment. It's going to be stabilized by a CD cluster deposits CD1. I'm sorry, CD8. Beg your pardon, CD8. So here we have an infected cell. Here's a nucleus right here. Here is a processed antigen right here on the MHC right there. This is the infected cell. Okay, all nucleated cells that have an MHC1. Now there's a T cell that has a receptor site on it for an MHC1. But that's a T cell right there that has a receptor site for an MHC1. But evidently the contact, the establishment, recognition, and attachment right here is not sufficient enough for the T cell to be activated. It has to be stabilized. It's going to be stabilized by a CD8. MHC1, CD8. MHC1, CD8. In a few minutes, I'm going to ask you to learn MHC2, CD4. CD4, MHC2. But right now, this is MHC1, CD8. MHC1, which is right here on the antigen presenting. There's a CD8 on the T cell that stabilizes its attachment. Very important that we have this cluster of deposits CD8. All right. Now, what happens after the activated T cell? After the T cell attaches to the infected cell. All right. Now, follow along right here. Activation of cell division right here. Now, the T cell is going to divide and produce a cytotoxic T and a cytotoxic memory cell. I don't care if you separate the cytotoxic memory cell. We talked about the types of T cells. Cytotoxic is one, helper T is one, suppressor is one, the memory cell is the fourth one. Okay, so the T cell fastens to a cell, a nucleated cell that has been infected, presented the antigen right there. The T cell fastens to it, and the CD8 stabilizes its attachment. That's where we are right now. So after this attachment has occurred, this activated T cell turns into a cytotoxic T cell and a memory cell. What does a cytotoxic T cell do? Now, cytotoxic is going to destroy, the cytotoxic T cell is going to destroy this target cell. This normally healthy cell right here that got infected in our body right there, presented MHC1, and it's a flag. Help me, help me, help me. Kill me, kill me, kill me. That's what's going to happen. So this activated T cell that becomes a cytotoxic T cell is going to kill this infected cell. It's going to kill it by 3 ways. Follow me on the chart right here. It's going to secrete perforins that form holes in the target cell and perforate right there. It's going to secrete cytokines that stimulate apoptosis. You may remember what apoptosis is. That's a programmed cell death, all right? Or it may secrete lymphotoxins that disrupt the cell metabolism. So 3 different ways that an activated T cell that is attached to an MHC1, stabilized by a CD8, 3 ways that this activated T cell kills this host infected cell. All right, that's what I want you to get out of this. This is a biggie. Now, let's take a look at MHC2. Remember, MHC2 is on the monocytes, the Kupffer cells, and the microglia cells, the fixed macrophages. So what happens when they engulf an antigen? All right, when they recognize and engulf the antigen, okay? So here we have an antigen that is engulfed by the antigen-presenting cells right here. Again, a part of the antigen fragment is presented to the cell surface on an MHC2. Now, along comes another T cell. Along comes another T cell right here, and it's got a CD4 on it. Evidently, CD8 does not recognize CD or MHC1. Let me back up. Evidently, CD8 does not recognize MHC2. Okay, 2 is recognized, stabilized by a T cell that has a CD4 on it. Okay, and that's just the way that it is. All right, now, what happens when a class 2 MHC on the antigen presenting cell is recognized by a T cell? All right, now, the T cell, as you take a look at it right here on figure 22-20 right here, the T cell right here becomes activated and forms a memory cell. I'm on the right-hand column right here. Forms a memory cell right here. And it's also going to release some cytokines that stimulate the B cell to produce antibodies. So evidently, the antigen presenting cell is not going to be destroyed by the 3 ways that we just looked at on the other, by the MHC1 CD8 complex. Evidently, the antigen presenting cell on the is not going to be destroyed. It's going to activate the T cell to become a helper T cell and a memory cell. Okay, that's what I want you to get out of this. The helper T is going to help produce the antibodies. That's very, very important. All right. Now, let's take a look at, that finishes up on MHC1s and CD combinations, okay? MHCs and CD1 and the CD combinations that we have right here. Now let's take a look at antibody production. Let's take a look at the structure and function of an antibody. Then we'll probably take a short break right here, and then I'll come back and I'll finish it. Don't overload the little box. Not want to overload the little box. Well, let's take a look at the structure of an antibody. Okay, you may know this from other classes, but an antibody to me has 2 long chains and 2 short chains. Okay? 2 long chains and 2 short chains. Now, there's several different types of antibodies, and there's 5 different classes in here. That's 22-1, table 22-1. I'm going to ask you to learn those. I'll help you with a shorthand version of what I want you to remember about each one of them, okay? Not all the information is right here. But anyway, we'll get to that in just a few seconds. But parts of an antibody right here. There's an antigenic determinant site. Antigenic determinant site right here. Antigenic determinant site. A-N-T-I-G-E-N-I-C-D-E-T-E-R-M-I-N-A-T-E site. But that's where it binds to the antigen. That's what determines that. I like the name of it. It tells me what the hell that it does. Okay? That is where the binding between the antibody and the antigen occurs. Okay. Very, very variable region that we have right here, because you will be exposed to 10,000 different antigens during your lifetime. 10,000 different antigens. And the hell of it is, you got an antibody already ready to bind to it. You really do. Already ready to bind to it. May not be many of them, but you got an antibody ready to bind to any antigen that you will be exposed to. A normal antigen. I had to do it that way. Now, there's another side on here. Remember back to complement activation. The classical pathway. Antibody-antigen interaction activates C1. Well, what I want you to know right here is the complement activation side. Complement activation occurs right here. And there's an SC region right here. FC region. Stands for crystallization fragment right here. It's an FC region, and that is a tissue determinant site. That's what I'm going to call it. That determines a certain types of tissue that it can bind to. And we'll take a look at some of the tissues, some of the antibodies present in mucus, present in tears or something like that. So it gives it a binding site to certain tissues, okay? The FC region. Those are the three regions of the antibody I want you to know, okay? Now, FC antigen determination. A hapten is rather interesting. It's an incomplete antigen. It's an antigen that does not elicit an immune response. Doesn't seem very fair, does it? The virus is really, really small. How come virus can, but some of these antigens can't? And we call these incomplete antigens a hapten right here. Now, steroids. I have three examples of haptens. Steroids, penicillin right here, and some of the short peptides. I don't have a good example for you on that. Short peptides, got to be shorter than a virus, so because the genetic material in a virus will elicit an immune response right there. But anyway, so steroids, penicillin, and short peptides are haptens themselves, okay? Now, let's take a look at one more, just for a brief minute right here. Let's take a look at the classes of antibodies. Table 22-1, classes of antibodies right here. I'm going to start on the left right here. IgG, most abundant. That's all I want you to write down. It's the most abundant antibody, the most abundant antibody right here. IgE, IgE, that is released by mast cells, okay? IgD is found in extracellular fluid, IgD, found in extracellular fluid right here. IgM is a pentamer. It's also the first class of antibodies that is produced. The first antibody that's produced when exposed to an antigen is IgM.